Our lab space in total in the College of Pharmacy here is approximately 5,000 square feet. And that's divided between this large open lab space. This is the most recent one, which was just finished, I believe, in 2019. And then the older lab space, which we started in originally, is approximately 800 square feet. So now we have, working in this lab space, we have 10 investigators, 10 faculty, and then we have a cadre of undergraduate students, master's student, and also PharmD students that come in and work with the PIs on their individual projects. This is Sarah. She's one of our third-year students, and she's working on an HPLC project, working on CBD.
We're looking for CBD and THC in the intestinal and gastric fluids, and how much is actually being extracted in the body.
Okay, but so she's using what's an HPLC system here, which works to isolate small molecules, so they can analyze the concentrations of small molecules. This is a flow cytometer, and this is the one I'm most familiar with. And this is the way to analyze single cells with multiple parameters. So I can analyze this, in this instrument presently, I can analyze 5 different parameters on a single cell at the rate of about 5 to 10,000 cells per second. So we can gather a large amount of data in a very small amount of time. As we move from the old into the new space, this space was originally designed by the research faculty in collaboration with architects to develop this open lab design, which hopefully fosters collaboration and interactions with faculty members. So each faculty member has at least one bay, individual bay. So we have our individual areas that we each have. Actually, for example, Dr. Glavy here has 2. And the idea being that this is where their research group do their work. And then we have common areas which are used by everybody or available to everybody. And an example of this would be a tissue culture lab here. So in this lab, we also do cutting edge molecular and cellular biology for cell analysis. And so for example, we have an imaging system here, which allows us to image gels when we analyze cells for DNA content or RNA content. Like Sarah was using in the HPLC, we have an FPLC in here for protein purification. We have a brand new ultracentrifuge for isolation of macromolecules. We have environmental chambers for growing various things. We can grow bacteria, we can grow yeast, algae also. Dr. Glavy grows algae. As we walk down further, we have an experiment going on analyzing nicotinic acid receptors, reading an individual cell, an oocyte, that has had the nicotinic acid receptor put into it, and then they're looking at its response to specific drugs. And you can see the tracing now. It's reading in real time the effects of the drugs on the cells. And so this is the end of our lab, and then we have at the very end, we have an office space that we have for students and postdocs that will allow them to be out of the lab if they need to do reading or research or writing as part of their job.